Good morning, students. The topic of today's discussion is gametogenesis. Before coming directly to gametogenesis, I would like to tell you that there are two modes of reproduction in organisms. One is asexual mode of reproduction and another is sexual mode of reproduction. Sexual mode of reproduction. As far as the organisms which reproduce through sexual mode of reproduction, they produce gametes. They produce gametes. Gametes are nothing but they are sex cells and they are haploid. The process of formation of gametes is called as gametogenesis. Process of formation of gametes is called as gametogenesis. One thing more I would like to add to your knowledge is that gametes, they are produced in specialized organs called as gonads. And these gonads, they are ovaries in female and testes in male. So males have testes as their gonads and females have ovaries as their gonads. The testes, they produce the male gametes they produce male gametes and the male gametes are called as sperms or spermatozoa. Whereas the ovaries, they produce the female gamete called as ova, in singular is ovum. Now the process of gametogenesis is, we can say, of two types. One is spermatogenesis and another is oogenesis. Spermatogenesis is the process of formation of male gametes. Spermatogenesis is the process of formation of male gametes, and oogenesis is the process of formation of ova or female gametes. So in other words, gametogenesis taking place in the male gonads is called as spermatogenesis, which results into the formation of sperms, and gametogenesis taking place in ovaries, which results into the formation of ova, is called as oogenesis. Now today I would be taking up the spermatogenesis. But before coming directly to the process of spermatogenesis, I would like to explain the structure of sperm or spermatozoa, because knowledge of the structure of spermatozoa is, I feel, essential for understanding the process of spermatogenesis, because during spermatogenesis the number of changes which take place for the formation of spermatozoa will easily be understood when we know the structure of spermatozoa. So what I have done is I have drawn a structure. I have I have I have drawn a structure. I have drawn a structure of the spermatozoa, and this is the structure of spermatozoa, and this is the spermatozoa of mammals. Now, if you look at the structure of spermatozoa, it consists of head, the middle piece, and the tail. Head consists of acrosome and the nucleus. Acrosome is the interiormost part of the spermatozoa, and the function of acrosome is it helps in the penetration of sperm into the ovum during fertilization. It dissolves the egg membranes, and nucleus, it contains what? It contains the chromosomes which are coming from the male parent, and the number of chromosome in the nucleus of the spermatozoa is half, that is, it is haploid. And just behind the nucleus we have the two centrioles. One is the proximal centriole and other is distal centriole, and these centrioles, they are arranged at arranged at right angles to each other. The distal centriole, it results in what? It results in the formation of we can say the flagellum, we can say the fibers which have the similar structure as those found in the flagellum or cilia. Then we have the middle piece. The middle piece consists of what? The middle piece consists of the thick fibers. Then we have the axial filament. Axial filament is 9+2 arrangement of the flagellum or cilium. It is similar to the structure of flagellum or cilium. The main function of this axial filament, the middle piece in the tail, is it helps in the propulsion of the spermatozoa in the liquid medium. Now, if you look at the middle piece, if you look at the TS of the middle piece, what happens is that these are the central fibers. Then we have the inner fibers, and the arrangement is akin, that is similar to those which are found where? In the flagellum or cilia of the protozoans. So this is 2 and these are 9 doublets. So 9+2 arrangement is seen where? In the middle piece, and besides 9+2 arrangement we have the 9 single or singlets, that is outer thick fibers, and these outer thick fibers, they are not present where? In the end piece, in the tail, in that tail region. Besides, in the middle piece we have also got the mitochondria. We have also got the mitochondria which are forming the mitochondrial spiral. The function of the mitochondria is they are the powerhouses, they are the energy houses. They provide the energy which is required for the movement, which is required for the propulsion through the liquid medium. Then this was just about the structure of the spermatozoa. One thing which you must be noticing is that most of the structures which are found in a normal cell, they are not present. They have been discarded. They have been discarded during the process of formation of the spermatozoa. During the spermatogenesis, what has happened is that most of the structures which normally are found where? In the cell, in the normal cell, they are not seen there. Like we have no Golgi bodies, as the Golgi bodies are converted into acrosome, or acrosome is derived from the Golgi bodies, which we will see where? In during the process of spermatogenesis. Now, thereafter I would be coming to what? I would be coming to the process of spermatogenesis. Now I hope it will be easier for you people to understand the process. Now, The whole process of the spermatogenesis, it is completed in two phases, I can say, or in two phases, that is formation of spermatid, formation of spermatid, one phase, and another phase is the formation of sperm or spermatozoa from spermatid. In that process is called as spermiogenesis. Now, just I would be describing, I would just be taking little time to make you understand the histology of the testis, as the spermatogenesis takes place where? In testis. And if you look at the structure of testis of the vertebrates, we find that the testis, it consists of, or it is formed of the tubules, the seminiferous tubules. The testis consists of seminiferous tubule, and this can be shown when we look at the TS of testis. When we look at TS of testis, testis consists of large number of similar tubules, and these similar tubules which constitute the testis, they are called as seminiferous tubules. Seminiferous tubules, if you look at the seminiferous tubules, they are formed of the fibrous sheath, and to the inner side of the fibrous sheath we have arranged at certain places the nursing cells, they are called as Sertoli cells. They are called as Sertoli cells. They are nursing cells, and in between the nursing cells we have arranged the germinal cells or germ cells, which are in different stages of the development. These which I am showing, which I am drawing with the help of what? With the help of red pen, or what? They are the germ cells. And these germ cells, they are in different stages, whereas we find the sperms towards the lumen. This is the lumen, and sperms, they are arranged towards the lumen. They are found towards the lumen. Now, the Sertoli cells, they are nurse cells, we can say. The germ cells which actually will be producing the spermatozoa. The spermatozoa, they are found where? In the lumen of the seminiferous tubules, and in between the seminiferous tubules. And what? And in between the seminiferous tubules we have got the connective tissue, and in the connective tissue are found the Leydig cells. Leydig cells. These Leydig cells are actually the endocrine. They form the endocrine part. They are what? Endocrine cells. They form endocrine part which secrete the male hormones androgen under the influence of luteinizing hormone coming from pituitary. Coming from what? Coming from pituitary. Now, as I have told you, as I have described this little about the structure of spermatozoa, the little about the TS of the testis, the structure of testis, and you might have got the idea till now that the sperms, they are produced They are formed where in the seminiferous tubules which constitute the testis. The whole process of spermatogenesis is categorized, is described in two stages. One is the formation of spermatid and other is spermiogenesis. Now if you look at the first phase, that is the formation of spermatid, formation of spermatid is the first phase, which again is subdivided into three phases, and these three phases are: one is multiplication, phase, second is growth phase, and third is maturation phase. Now, as we know that the germ cells are present where in the seminiferous tubules, and the germ cell which undergo multiplication, they are called as primordial germ cells. Primordial germ cells. The primordial germ cell undergoes the repeated mitotic divisions, divisions. So this is the PGC, primordial germ cell. It undergoes repeated mitotic divisions and leads to the formation of sperm mother cell. Leads to what? The formation of sperm mother cell. Leads to the formation of sperm mother cells, also called as spermatogonia. Sperm mother cells, they are also called as spermatogonia. So the spermatogonia are formed as the result of the repeated cell divisions, and this repeated cell division is mitotic in nature. That is, the number of chromosomes in primordial germ cells is 2n, and again 2n, and again 2n. When the multiplication stops, at the end of the multiplication, what is formed is the sperm mother cells. So the whole of the process is the first subphase, that is multiplication phase. Now the sperm mother cells, they undergo the growth phase. They increase in size. They do what? They increase in size. They grow in size, and the size become exactly double that of the sperm mother cell. So we can say that during this phase, growth takes place. So this is growth phase. At the end of growth, what happens is that sperm mother cells or spermatogonia, they undergo the growth, they increase in size, the size become double, and at this stage we get the primary spermatocyte. And still, the number of chromosomes is diploid, that is 2n. Total number of chromosome is 2n. It is diploid in nature, primary spermatocyte. In the primary spermatocyte, it undergoes what? The third phase. The third subphase, that is maturation. And during maturation, what happens is meiosis takes place. Meiosis. The primary spermatocyte, it undergoes meiosis. It forms what? It undergoes meiosis first. This is meiosis first. Meiosis first, which actually is the reductional division. So the number of chromosomes found were in the secondary spermatocyte. Secondary spermatocyte is in, you might be knowing, you would be having a knowledge of what? The knowledge of meiosis. So this is the first meiotic division, is the reductional division. Now these secondary spermatocytes, they undergo what? They undergo meiosis second. Meiosis second, which is, you very well know, is the equational division. And what happens is that equational division, and at the end of meiosis second, the cells we get is the spermatids. The cells we get is the spermatids, and all of them, they are haploid. One thing which we came across is that the formation of spermatid is not the end point of the spermatogenesis, because still the spermatid is like any other cell. It has a round shape, it has the unnecessary cytoplasmic bodies which are not at all involved, which are not at all required for which are not at all required for the fertilization. So then comes the second phase, and this second phase is the spermiogenesis. Spermiogenesis. So during spermiogenesis, what happens that a large number of changes take place where in spermatid, large number of changes take place where in a spermatid, this is spermatid, large number of changes take place in spermatid to form the sperm. So you can very well imagine the changes which take place when during the spermiogenesis, that the rounded, that a ball like cell, it changes into the spermatozoa. What are the changes which take place during spermiogenesis is that extra cytoplasmic bodies are eliminated. That is, cytoplasm is last Golgi complex, it results into the formation of very important structure called as acrosome. From the nucleus or in a non-histone proteins are lost, and this is all done for reducing the weight, reducing the weight, so that extra energy is not used for the transportation, for the movement. And then we have the changes